A very good morning to all of you. Today I will be talking about a topic, hemostasis. What is hemostasis? What do you mean by hemostasis? You can split the word hemostasis into heme as well as stasis. What is heme? Heme means blood. Stasis means to stop or to halt or to arrest. So hemostasis, in a simple words you can say hemostasis is the arrest of bleeding. So coming to the introduction, how do you define hemostasis? Hemostasis is defined as the process of arrest of bleeding while maintaining blood in a fluid state within the vascular system. Hemostasis is defined as the process of arrest of bleeding while maintaining the blood in a fluid state within the vascular system. Hemostasis is the mechanism by which loss of blood from vascular system is prevented by a complex interaction of vessel wall, platelets and plasma proteins. So hemostasis is the arrest of bleeding while maintaining the blood in a fluid state. This is a mechanism by which the loss of blood from the vascular system is prevented by a complex interaction of vessel wall, the platelets and plasma proteins. The role of platelets is very important in hemostasis. I'll come to that later. And hemostasis is actually divided into two stages. Hemostasis happens broadly in two stages, mainly the primary as well as the secondary. So primary stage as well as the secondary stage are the two categories or two stages which you can broadly divide the hemostasis. What is primary hemostasis? Primary hemostasis is the initial stage. This is the initial stage during which vascular wall and platelet interact to limit the blood loss from damaged vessel. So the role of vascular wall and the platelets are very important here to interact to limit the blood loss from the damaged vessel. This is also called as temporary hemostasis, as the platelet plug alone can't stop bleeding for a longer duration. So it's temporarily a plug is formed, temporarily a clot is formed. It cannot stop bleeding for a longer duration. So it's known as temporary hemostasis. What is secondary hemostasis? What is secondary hemostasis? A stable fibrin clot is formed from clotting factors by enzymatic reactions. This is also called as definitive hemostasis, as the formation of clot in the vicinity of platelet plug finally stops bleeding. So here there is a stable fibrin clot is formed from clotting factors. The clotting factors, there are 13 clotting factors. Separately it can be asked. Name the clotting factors as a long essay it can be asked. So there are 13 clotting factors plus extra there are other factors also. Among the 13 factors, factor 6 is absent, and you should know the name of different factors. That's a separate thing you have to know. It's not there in this PPT, but it's a separate topic. Later I'll come to that. So a stable fibrin clot is formed from clotting factors by enzymatic reactions. This hemostasis is also called as definitive hemostasis, as the formation of clot in the vicinity of platelet plug finally stops bleeding. So what is temporary hemostasis, and the second one is definitive hemostasis. Although formation of blood clot is needed to arrest blood loss, ultimately blood clot needs to be dissolved to resume the normal blood flow, and the process of dissolution of clot is called fibrinolysis. So what do you mean by fibrinolysis? The process of dissolution of clot is called fibrinolysis. When it is actually happening, the formation of blood clot is needed to arrest blood loss. So what happens? The blood clot needs to be dissolved to resume the normal blood flow. So this process of dissolution of clot is called as fibrinolysis. Bleeding occurs when the blood vessels are damaged. The vascular injury initiates a series of events in sequence that finally forms a clot at the site of injury and prevents the loss of blood from the body. Thus, hemostasis is a life-saving process. It is a life-saving process that maintains the homeostasis of blood volume. Every time what happens here is the students get confused with hemostasis and homeostasis. Both are different. I have already taken the class about homeostasis. It is the maintenance of the stable environment. It's the maintenance of the internal consistency of the environment. Hemostasis is a small one-letter difference actually. Homeostasis and hemostasis, but the meaning itself is a wide difference comes in the meaning. So what usually we see in the exams as they confuse homeostasis and hemostasis. Just see the word properly and then write in the exam. Hemostasis is arrest of bleeding. Homeostasis is the first topic what we learned in general physiology. Okay. So both are different actually. So don't get confused with hemostasis and homeostasis. So hemostasis is a life-saving process that maintains the homeostasis of blood volume. So hemostasis is the arrest of bleeding. There are two categories mainly, primary and secondary. Primary is also known as temporary hemostasis, and secondary is also known as definitive hemostasis. So actually there are three steps of hemostasis. There are three steps: vasoconstriction, formation of temporary hemostatic plug, and formation of definitive hemostatic clot, or also known as blood coagulation. So this is what I told you, steps of hemostasis. Steps of hemostasis, there are mainly three steps of hemostasis: vasoconstriction, formation of temporary hemostatic plug, and the last one is formation of definitive hemostatic clot, also known as blood coagulation. Coming one by one, first stage is vasoconstriction. Vasoconstriction is the immediate response of the blood vessel to injury. When exposed to injury, the first response, the immediate response of the blood vessel is vasoconstriction. It occurs due to the contraction of vascular smooth muscle. So when there is a response to injury, when the injury is exposed, what happens is the immediate response of blood vessel is the vasoconstriction, where the contraction of the vascular smooth muscle takes place. So instantaneously it stops blood loss and also facilitates platelet plug formation. Contraction of vascular smooth muscle to injury is initially a mechanical response, which is also known as stretch-induced contraction. Later, the vasoconstriction is maintained and potentiated by the secretion of vasoconstrictor substances like serotonin from the activated platelets. So when there is an injury, the immediate response, what you see is vasoconstriction, where there occurs due to the contraction of vascular smooth muscle. It instantaneously stops the blood loss and also facilitates the platelet plug formation. So, and later the vasoconstriction is maintained and potentiated by a series of a list of or a secretion of vasoconstrictor substances, especially like serotonin. That is about vasoconstriction. Second stage is formation of temporary hemostatic plug. So, formation of platelet plug at the site of injury is called temporary hemostatic plug. This occurs due to three properties of platelets. Mainly the action of platelets or function of platelets you can remember as 3 A's. You can remember as 3 A's: adhesion, activation, and aggregation. The temporary hemostatic plug occurs due to three properties of platelets, mainly adhesion, activation, and aggregation. So 3 A's you remember: adhesion, activation, and aggregation. The initial response of platelet to vascular injury is the change in the shape of the platelets and also its increased surface adhesiveness to the injured vascular endothelium. This is called adhesion. As I told you, there is adhesion, activation, and aggregation. So initial response of platelet to vascular injury is the change in the shape of the platelets, and it increases the surface adhesiveness. So there is known as adhesion. Platelets also stick to each other, that is known as aggregation. It sticks to each other, or sticks like one platelet sticks to the other platelet at the site of the injury. Platelets also stick to each other or aggregate at the site of injury. This is called as aggregation. Simultaneously, platelets are also activated to release or secrete a number of chemicals. Release reaction or secretion is also known as that further facilitate vasoconstriction, adhesion, and aggregation. The whole process finally results in the formation of a platelet plug that arrests bleeding temporarily. As the platelet plug is not a stable one, the plug is also called as temporary hemostatic plug or primary hemostatic plug, and the process is called as temporary hemostasis. So primary hemostasis or temporary hemostasis, actually it is the formation of a temporary hemostatic plug, where you can see the action of platelets, mainly the function or action of platelets can be remembered as 3 A's: adhesion, activation, and aggregation. After aggregation, there is also something known as release reaction or secretion. So this all results finally in the formation of a platelet plug that is just for a temporary time. So it is also called as temporary hemostatic plug or primary hemostatic plug. The last stage is blood coagulation, which is also known as formation of a definitive hemostatic plug. That is a secondary hemostasis. So what happens here? Immediately following injury, the clotting mechanism of blood is initiated. This results in the formation of blood clot at the site of injury that stops bleeding. The formation of blood clot occurs in and around the platelet plug, and the plug formed is a stable one. As the plug formed is a stable one, this plug is also called as a definitive hemostatic plug or secondary hemostatic plug. Secondary hemostasis is also known as definitive hemostasis. Is nothing but the blood coagulation step. This process is also called secondary hemostasis, as a step follows a primary hemostasis. That's what I told you. You can see a flowchart here which shows the mechanism of hemostasis. This flowchart I have taken from GK Pal. So this is a summary of what I have told you now. So you can see when there is a vascular injury, what happens when there is a vascular injury? When there is exposed to injury, what happens? You can see there can be a collagen exposure here. You can see there can be an immediate response. The immediate response after vascular injury can be vasoconstriction. So mechanical vasoconstriction can take place, which can directly cause temporary hemostasis, which can directly form temporary hemostasis, or which can directly result to temporary hemostasis. So when exposed, when there is a vascular injury, either it can have a mechanical vasoconstriction, it can have a collagen exposure. Also you can see one more thing is, it can either release the tissue thromboplastin. Okay, so when there is a collagen exposure, you can see the 3A's of platelet, the functions of platelets, mainly the 3A's: adhesion, activation, aggregation, and also release and secretion, release, reaction and secretion. So when there is a collagen exposure, platelet adhesion and aggregation takes place. This also results in platelet activation and release. After the release action, you can see a formation of temporary hemostatic plug. It's a short form THP. THP is a temporary hemostatic plug. So THP is formed here, temporary hemostatic plug is formed here, which results in temporary hemostasis process. So if there is a release of tissue thromboplastin, when there is a release of tissue thromboplastin as a part of injury, you can see there is an activation of clotting. This can result in definitive hemostatic plug formation, that is also known as DHP formation, which is written as a short form here, which results in permanent hemostasis or blood coagulation. So when either there can be mechanical vasoconstriction that directly can cause temporary hemostasis, or it can be collagen exposure, which can result in activation, which can result in platelet adhesion, aggregation, activation, release reaction, which can form a temporary hemostatic plug, which can result in temporary hemostasis, or it can release of tissue thromboplastin can also takes place as a part of vascular injury, which results in activation of clotting factors, and it can lead to blood coagulation or definitive hemostatic plug formation, which is our permanent hemostasis. So these are the different mechanisms of hemostasis. It's in a flow chart. If you study this flow chart, you will have a rough idea, but you have to explain. If it's a 10 marker questions, you have to explain separately. Vasoconstriction you have to explain, formation of temporary hemostatic plug you have to explain. Also you have to explain about the formation of definitive hemostatic plug or blood coagulation. That was all about mechanism of hemostasis. Now coming to the role of platelets in hemostasis. As you saw there, what happens after the vasoconstriction when there is a temporary hemostatic plug, the action of platelets comes into takes place. The role of platelets comes into action. So role of platelets in hemostasis. The role of platelets in hemostasis following injury to blood vessel, following a vascular injury. What happens to platelets? There are three actions or three functions of platelets. As I told you, adhesion, aggregation and activation you can see. Following injury to blood vessels, the platelets aggregate at the site of injury and seal the opening in the vascular tree. This is the first and important step in prevention of bleeding, which is called temporary hemostasis. So this is the first and important step in prevention of bleeding, which is called temporary hemostasis. Simultaneously, mechanisms of coagulation of blood are also activated that form blood clot. The process is combinedly known as definitive hemostasis. So same thing what I have told earlier, formation of temporary hemostatic plug as well as formation of definitive hemostatic plug is one of the important functions of platelets in hemostasis. So, though platelet is essential for temporary hemostasis, it also contributes to blood coagulation and clot retraction. Blood coagulation is also known as definitive hemostasis, and clot retraction is something also known as clot retraction. So clot retraction is a separate thing. It's not a blood coagulation is different, clot retraction is different. Platelet also controls thrombolysis. So blood coagulation, clot retraction and thrombolysis are also the functions of platelets. Even it can be asked as 3 markers, functions of platelets, list out functions of platelets. It can be asked directly. For any course it can be asked, MBBS, BDS, BPT, BSc Tech Nursing, anything. Okay, so platelet functions is very important. You have to know it properly. So I am not talking individually about platelet functions. As a part of hemostasis, the role of platelets is what I am talking about here. So one of the functions, blood coagulation is there, clot retraction is there, thrombolysis is also controlled by platelets. Immediate hemostasis following vascular injury is achieved by formation of platelet plug. It is also called as platelet thrombus. So platelet thrombus is formed at the site of injury when there is a vascular injury as platelet thrombus can be washed away unless supported by a clot. This is called temporary hemostatic plug. Temporary hemostatic plug formation depends on adhesion, platelet aggregation and release reaction. So coming one by one, what is platelet adhesion? Platelet adhesion. The damage to blood vessel exposes underlying portion of the vessel wall that are normally concealed from the circulatory platelets by intact lining of the endothelium. The platelets have high affinity to adhere to the exposed vascular, which is called as platelet adhesion. Especially exposed collagen, as a part of injury, the collagen is exposed, and even there is a factor known as von Willebrand factor in the injured vessel wall attract platelets, as platelets have receptors for them on their cell membrane. The platelets have receptors for the exposed collagen and the von Willebrand factor, so they attract each other, and it results in the platelet adhesion. So platelet adhesion is immediately followed by the platelet aggregation at the site of injury. So that's about platelet adhesion. The second one is platelet aggregation. Fibrinogen, glycoprotein 2B and 3A, that's GP, 2B and 3A, and thrombin promote platelet aggregation. So fibrinogen, glycoprotein 2B, 3A and thrombin promote the platelet aggregation. Fibrinogen, glycoprotein 2B, 3A and thrombin promote platelet aggregation. Simultaneously, platelets are activated and activated platelets release the chemicals from their granules. These chemicals, especially ADP and platelet activating factor, that is known as PAF, ADP and PAF further facilitate the platelet aggregation. So the chemicals which is released as a part of activation of platelets, mainly the ADP and platelet activating factor, further facilitate the platelet aggregation. Thus, accumulation of more platelets at the site of vascular injury facilitates the process of temporary hemostatic plug, that is a platelet thrombus formation. Then the formation of clot is initiated. Fibrinogen, glycoprotein 2B3 and thrombin promote the platelet aggregation. As a result of an injury, the platelets are activated simultaneously, and the activated platelets release the chemicals from their granules, especially the ADP and platelet activating factor, which further facilitates the platelet aggregation and forms a platelet thrombus. From there, it is the formation of the clot is initiated. You can see a diagram here, the role of platelet in hemostasis. A, B, C is given. First one is platelet adhesion. This first A is the platelet adhesion. What is happening here? What is all about platelet adhesion at the site of vascular injury, and you can see here the exposed collagen in damaged endothelium here. So as a part of vascular injury, the collagen is exposed in the damaged endothelium. So it promotes the adhesion. You can see the second one, what happens is platelet aggregation. So the second one is platelet aggregation. So you can see here platelet is aggregating here. Okay, the first one is the injury, there is exposure of the collagen in the damaged endothelium. The second you can see platelet aggregation. The third diagram you can see the C, what is formation of a fibrin threads around the aggregated platelets. So you can see just can you see the mouse here, what I am pointing this where there is an injury here. When the injury is happening, there is an exposure of collagen in the damaged endothelium. So there is a site of injury, you can see site of injury arrow, it is marked. The second one is endothelium, exposed to collagen, you can see green color, that is exposed to collagen in the damaged endothelium. So there what happens here is you can see the platelet adhesion taking place at the site of injury. The platelets are adhered. This red ones are the platelets, the green is exposed to collagen. This is the site of injury, you can see a gap here. So these platelets are the platelets adhesion takes place in the site of injury. The second picture you can see the platelets get aggregated. See, they are like a grapes, you can see it's like aggregated here. So a platelets are getting aggregated here, which forms the platelet plug. See the last one, the platelets aggregated here is what happens, it is surrounded, or you can see the blue mark here, which is fibrin thread. So like a thread it is formed, the fibrin threads in and around the aggregated platelets can be formed, can be seen here in third stage, that is a definitive plug formation. So there later a clot, you can see a fibrin thread itself is formed here, that is the end of the hemostasis. So there is a definitive hemostatic plug or blood coagulation have taken place here at the end of this third diagram, you can see. So this is about the picture, platelet adhesion, platelet aggregation and platelet, the formation of the fibrin thread in platelet plug. So these are the role of platelets mainly. Similarly, this is one more diagram, you can see the synthesis of thromboxane A2 that promotes platelet aggregation. There is one more thing happening in platelet aggregation process. There is nothing but the Thromboxane A2 is synthesized. Membrane phospholipids, arachidonic acid. You can see phospholipases and cyclooxygenase inhibition by steroids in this process. In this step, inhibition by aspirin, other NSAID. You can see prostaglandin G2, prostaglandin S2 with the help of thromboxane synthase, it converts to thromboxane A2 and results in promotes in platelet aggregation. So here NSAID is a painkillers which usually we use non-steroidal and inflammatory drugs. So cyclooxygenase inhibition by aspirin, other NSAID, phospholipase inhibition by steroids. So you can see here at last the thromboxane A2 is synthesized which can promote the platelet aggregation. So one more function of the platelet function was platelet activation and release reaction. Adhesion was over, aggregation was over. Now the last one is the activation and release. So adhesion of platelets to damaged vascular endothelium activates the platelets. Polymerization of microfibrillar actin occurs in activated platelets that results in pseudopodia or spicules formation. Platelets are also activated by tissue factors and chemicals released from the granules of the platelets, especially PAF. I told you ADP and PAF, platelet activating factor, are the chemicals secreted as a part of activation of platelets. Thrombin and ADP also promote activation. So again, it is the same thing is coming. ADP and PAF are the main factors which is released as a part of activation. Even you can see thrombin also along with ADP and PF. So especially you have to remember the factor PAF, that's platelet activating factor. The activated platelets release various chemicals from their granules to the exterior via the canalicular system. The process is also called as release reaction or platelet release. That is release reaction and secretion. So release reaction, release means the chemicals are released, especially the ADP, PAF and thrombin, and there is a secretion, that is a platelet release is known as a secretion. So these are the different actions or the roles of platelets in the hemostasis. So this is a table showing the different factors that promote as well as inhibit aggregation and activation of the platelets. So factors promote aggregation and activation. You can see a list of factors: ADP, platelet activating factor, thrombin. As I told, these three factors already. You can see also serotonin, thrombospondin, fibrinogen, von Willebrand factor, immune complexes, plasmin, tissue plasminogen activator. Damage to the vascular endothelium directly can promote the aggregation, slowing of blood flow. So these are the different factors that can promote aggregation and activation. What are the factors that inhibit aggregation and activation? When the blood flow is very fast blood flow, prostacyclin, nitric oxide, or also known as EDRF. EDRF is the endothelium derived relaxing factor. Also, the inhibitors of thrombin generation and action can inhibit the aggregation and activation of the platelets. Now, what about the other hemostatic functions? Role in blood coagulation. The role in blood coagulation. Activation of platelet leads to increased platelet coagulant activity. The platelet factor 4, PF4, acts as a cofactor for blood coagulation. Platelet synthesizes coagulation factor 5 and 11. There are 13 clotting factors, as I told you. I didn't name each one individually, but there are 13 clotting factors and the factor 6 is absent. So platelet synthesizes coagulation factor 5 and 11. The platelet phospholipids present on the platelet surface accelerate the formation of 5A, 8A and 11A. The 5A is essential for the conversion of prothrombin into thrombin. So there is a step where the prothrombin is converted into thrombin by the factor 5A, that is activated factor 5. They also facilitate the conversion of 10 to 10A and prothrombin to thrombin. So the activation or the conversion of factor 10 to activated factor 10 and the prothrombin to thrombin is facilitated by the hemostatic function, mainly the role in the blood coagulation of platelets. Also, platelets accelerate thrombin formation. Okay, they facilitate the conversion of 10 to 10A and prothrombin to thrombin. Role in clot retraction. If platelets are present in the clot in a test tube, within minutes to hours, the clot contracts, extruding a very large fraction of serum. This is called as clot retraction. The platelets present in the clot in a test tube, within minutes to hours the clot can contract, which results in extruding a very large fraction of serum, which is called as clot retraction. The platelet filopodia extends into fibrin clot and fibrin strands tug with filopodia. The platelet filopodia extends into fibrin strands tug with the filopodia. The shrinkage of platelet with contraction of filopodia causes internalization of fibrin that causes clot retraction. So there is something known as filopodia and the internalization of fibrin takes place, which results in clot retraction here. Fibrin is a thread, a clot you can say. When you define clotting time, the time taken from the onset of bleeding till the formation of fibrin thread, that is a clot, is called as clotting time. So when you take it as a clot or something, it is a fibrin thread forming at the end. That is known as the clot. So here the fibrin strands tugged with filopodia here. The platelet filopodia extends into fibrin clot and the fibrin strands tugged with filopodia. This results in the shrinkage of the platelet with contraction of the filopodia, causing the internalization of fibrin that causes clot retraction. One of the functions of platelet is clot retraction. In vivo, the phenomenon akin to the clot retraction is the consolidation of the thrombus, which not only firmly seals the opening in the injured vessel but also facilitates the wound healing. So one more important function you can see is facilitating the wound healing. The clot retraction also prevents the process of thrombolysis. Platelet deficiency prolongs the clot retraction time. You can check the clot retraction time. In this, what happens, the platelet deficiency can prolong the clot retraction time. More important function is the role in the thrombolysis. So mainly three important functions you have to keep in mind: blood coagulation, clot retraction, and thrombolysis. Thrombospondin, TPA, tissue plasminogen activator, and plasmin. The short form of tissue plasminogen activator is TPA that are pro-fibrinolytic, are synthesized and released by platelets. Antifibrinolytics like plasminogen activator inhibitor 1 and A2 antiplasmin are also secreted from the platelets. Antifibrinolytics like plasminogen activator inhibitor 1 and A2 antiplasmin are also secreted from the platelets. Platelets, by causing clot retraction, decreases the efficiency of thrombolysis. Antifibrinolytic effects of platelets predominate over their pro-fibrinolytic effects in vivo, as platelet-rich thrombus is known to resist thrombolysis. So the role in thrombolysis, mainly the thrombospondin, tissue plasminogen activator, and the plasmin are pro-fibrinolytic, are synthesized and released by the platelets. So antifibrinolytics like plasminogen activator inhibitor 1 and A2 antiplasmin are also secreted from the platelets. So there are two things here: pro-fibrinolytic as well as antifibrinolytic. So here the antifibrinolytic effects of platelets predominate over the pro-fibrinolytic effects in vivo, I am saying, and as the platelet-rich thrombus is known to resist thrombolysis. That's the reason why the two effects, mainly antifibrinolytic effects, predominate over the pro-fibrinolytic effects. You can see the role of blood vessel in hemostasis. The last slide I am talking today is about the role of blood vessel in hemostasis. You can see the different factors that favor hemostasis as well as that inhibit hemostasis in the endothelial cells. So this is a picture which shows the endothelial cells. What happens in the endothelial cells? There are factors which are favoring hemostasis as well as which are inhibiting the hemostasis. So the list of factors here which favors hemostasis are vasoconstriction, the synthesis of tissue factor, the von Willebrand factor, and the PAF, platelet activating factor. So these are the factors which favors the hemostasis, whereas the factors that inhibit hemostasis are synthesis of thrombomodulin, protein S, prostacyclin, TPA, that is tissue plasminogen activator, and heparin-like substances. So these are the substances that can inhibit hemostasis. So the role of blood vessel in hemostasis, you can see in the endothelial cells, you can see the factors favoring hemostasis as well as inhibiting hemostasis. So this is all about hemostasis. Hemostasis means arrest of bleeding. There are primary hemostasis and secondary hemostasis. The primary hemostasis is also known as temporary hemostasis. The secondary is also known as definitive hemostatic plug formation, that is the permanent hemostasis. So the three steps are vasoconstriction, formation of temporary hemostatic plug, as well as the formation of definitive hemostatic clot or blood coagulation. So there are important, the role of platelets is very important here. Mainly there are three actions, three A's of platelet you have to remember: platelet adhesion, platelet aggregation, platelet activation. Also you can see is release, reaction, secretion of certain chemicals like ADP, the platelet activating factor, etc. So there are list of factors that can inhibit and favor the hemostasis. So one by one I have told you about the platelet action, the role in blood coagulation, the role in clot retraction, the role in the thrombolysis. So this can be asked as define hemostasis and explain the mechanism of hemostasis. It can be asked as a 10 markers, it can be asked as a 5 markers, or any short answers can be asked in this topic, like functions of platelets, etc. So MCQ, anywhere it can come. So it's an important topic coming in physiology. So this is all about hemostasis. So I'm stopping here. I'm winding up for today. Thank you.